Our speaker today is Axel Scherer, who is a distinguished visiting professor at the Thayer School of Engineering. Axel is the Bernhard Neches Professor of Electrical Engineering, Applied Physics, and Physics at Caltech, as well as a visiting professor here at Dartmouth. He received his PhD in 1985 from the New Mexico Institute of Mining and Technology, and after that he worked in a microstructure research group at Belcor. He joined the electrical engineering option at Caltech in 1993. Professor Scherer's group now works on micro and nano fabrication of optical, magnetic, and fluidic devices. He has co-authored over 300 publications and holds over 70 patents in the field of optoelectronics, microfluidics, and new nanofabrication techniques. I would like to ask you also to refrain from using laptops out of courtesy for our speaker. Please welcome Axel Scherer.
Thank you very much, Alex, for this nice introduction. I really appreciate it. Today, what I'd like to do is give you sort of an overview of what we're doing in terms of miniaturization. The work that I do with students in my lab involves miniaturization of devices and systems. And many of the applications range from medical applications all the way to quantum physics. And I'd like to talk about some of the things that we do and how we do them, focusing on more the technique and the technology than the application. The idea really is to give you an overview of what we do. So actually, I should certainly acknowledge the people that do most of the work. That's the students in my group. They build the systems, vacuum systems, that allow us to build the devices that I'll talk about. They do the work. They're the ones that actually come up with most of the ideas. And my job is to harness these ideas and build systems. So we take advantage of a legacy of lithography. I'm going to talk about how lithography allows us to miniaturize things and how lithography allows us to integrate things. But lithography is an exercise that's not tremendously new. It has allowed the electronics industry to, within the last 50 years or so, go from single devices to integrated chips. And other technologies are rapidly following suit. So miniaturization through lithography is something that isn't limited to silicon. Any other material you can derive the same capabilities, usually the ability of making more complex systems and making things smaller. And we're moving away from the traditional assembly process in these small sizes to a process of printing. So instead of assembling optical systems, I'll show you that we print them. And they're aligned because we print them with appropriate masks that allow the optics to work. This, of course, allows us to make things cheaper, makes things smaller, more compact, more robust, and so on. Printing, of course, is something that's been developed over the ages since the medieval times of Gutenberg. The reason he printed books was because he wanted to make them cheaper, more accessible. So instead of transcribing things, he printed them. And ever since then, we've been doing this, just at smaller scales. More resolution, better instruments. But the concept is still the same. We're printing things, many things at the same time. And it saves us cost. And of course, the microelectronics industry has taken this, and there's a rule now called Moore's Law that we're all familiar with, where the complexity of systems is constantly increasing on this geometric scale. All right. This is interesting because it's not limited to electronics. We can think about, first of all, the reason why electronics does this. then we can translate it to other applications. So one of the reasons why the electronics industry does this printing and integration is because it's a sort of a real estate transaction. There's a certain amount of money you pay for a square centimeter of integrated circuit. That cost has not changed over the last decades. The cost is somewhere between $5 and $10 per square centimeter. It's the same cost that it was in 1971. The interesting thing is you can start adding more and more function to that square centimeter, and then you can sell it for more money. And it turns out that that's a good business model, and that causes us to make more and more complex devices. The other reason is it's difficult to move data from chip to chip. You have to put it into bond pads or bump pads or translate it some way from the chip to something else, and you may lose information. So you want to do as many things as you can on the same chip. Well, it turns out that's the same paradigm that you want to use for making biological systems or medical diagnostic tools. You don't want to lose molecules in this case rather than bits. Okay, so I'll talk about different technologies. I'll first talk about optoelectronic integration, then I'll talk about microfluidic systems, then I'll talk about integration of different systems together through lithography, talk a little bit about the pathology instruments that we're building today, and then I want to go to the smallest scale where we can control things by geometry. So we start with silicon. Silicon is a wonderful electronic material. Many of you know that. Some of you probably also know that it's a wonderful optical material. In the visible light, of course, it's absorbing, but in the near infrared at 1.5 microns, you have the silicon becomes completely transparent. It's actually one of the most transparent materials where absorption only occurs on surfaces. And you end up having this beautiful opportunity of making something that's very transparent but has a high index of refraction. And this observation led to the idea of building little circuits made out of silicon, optical circuits in this case. Well, the enabling technology that allowed us to do that maybe 15 years ago was the development of a new process called silicon on insulator. Basically, wafers are made by bonding crystalline silicon on top of a handle of silicon that has an oxide on it. What that allows you to do is to decrease the leakage currents from circuit element to circuit element. What it also allows you to do is to build waveguides. You can actually take this wafer, this SOI wafer, which has crystalline silicon on top of oxide, and you can cut it up into different geometries. You can make structures like this. You can make thousands of little optical elements that can be integrated in such a way that they talk to one another. This is by printing. You can make optical and electronic elements next to each other. And you can go further, you can make systems like this, chips, that have hundreds of thousands of transistors to run optical elements. And you can buy these things now. So Luxtera is a company that we started early on in 2001. Now they can sell you these things. It's basically allow you to do optical communications. You can buy a USB cable. It doesn't have to work at a couple of megahertz. It works at 40 gigahertz. So it allows you to do very fast data communications. Okay, so this is a mature technology. But what's also possible is you can go further. You can build devices that work far, far beyond what electronics can give you today. You can make switches like this, which can work at terahertz frequencies, optical switches, things like that, because you can print these components on the silicon, and you can add then the nonlinear material on top of them. Okay, so there's great opportunity in printing and then modifying things. Well, I'm particularly interested in the area of medicine. And so one of the thoughts, nice ideas, is why don't we take this technology, which allows us to have waveguides printed on a silicon wafer, and convert these in some way or another to a sensor that allows me to tell whether I have certain protein level. Well, in order to do that, what I need to do is I have to build an optical filter or something that measures something. So let me say I'm measuring the refractive index. If I have some bonding event, the refractive index will go up. I can build a resonator simply by printing a ring like this. And then I can coat this resonator with some material, maybe antibodies or whatever binding material, some glue. And then I observe the shift of this resonance as a function of time, and it can tell me whether I have bonded something on the surface. Well, it turns out that resonators don't have to look like round things. They can be like this. This is actually a beautiful resonator. It's actually a wire, in this case a waveguide, and a wire, and a resonator at the same time. You can actually capture light in here, hold it, and a Q of this kind of resonator can be we've made them with 200,000 Q. Very small structure. You can put current through it to heat it up. You can do all sorts of neat things with this. You can coat it. You can make a laser out of it. I'll show you in a moment. You can do all sorts of very nice things. And it is essentially a printed structure. It's made by simple lithography. Well, that brings me to the point of how do we do something like an optical experiment on such printed structures. We need to have some fluids delivery system that allows us to put the sample next to this structure. That leads me to fluidics. Well, in fact, you can do the same thing that we did lithographically defining electronics or optics. You can also do it with fluidics. In this case, we use a technique that's called replication molding. So we actually print some mold, and then we replicate an elastomer on top of it. Looks something like this. You have the mold here, you pour the elastomer in there, you cure it for a while, and then you peel it off, and you put it on your new substrate. It's essentially a lithographically enabled replication mold. You can build systems like this by using replication molding. These are molded channels that are controlled with molded pneumatic systems that can allow me to control material within 1 picoliter to move it around on a chip. All right. In this case, I make a mixer. There's food dye in these channels, and I mix together different food dye. Okay. But you can do many other things. Okay. Here's when it starts mixing. The neat thing here is you can make very complicated fluidic systems by aligning printed replication molded structures. Now, if I take these replication molded structures that allow me to move picoliters of material around, and I build them together with my printed optics, I can start building spectrometers. Okay. So this is a spectrometer. Actually, it's a 3-wavelength spectrometer. This is a photonic crystal laser. Each laser element looks like this. You can maybe see this one here. It's a laser that's sort of an interesting laser in that it has a hole in the middle of it. It's deliberate. That hole is my cuvette. I'm going to put material in it. But it's a laser, and so light has to interact with the material that is in my hole. It's actually, geometrically, it's only a half a wavelength long, so it has to be in there. And so basically, I've built an integrated system, and in this case, I built 3 of them next to each other, each one working at a different wavelength. All right? So it's not a tremendously high-resolution spectrometer. It's a 3-wavelength spectrometer, but you can easily make this longer, make many more. Then I can take these lasers that are these structures with little holes in them, and I dip it into a solution, and when the thing lases, it tells me what's inside of the little hole in the middle. And in fact, I can see the difference between methanol and isopropyl alcohol or water by simply looking at the laser wavelength, the laser coming out. All right? And I'm measuring something on the order of 10 to the minus 18 to 10 to the minus 17 liters, because that's the mode volume of my laser. Okay? In fact, this is what the mode looks like. It's concentrated right in this hole in the middle. Okay? I can even trap things. I can have little nanoparticles. They come by, they get optically trapped. optical tweezing holds them in place. This doesn't have to be the only geometry. As I said before, you have these beautiful wires, these photonic wires that basically can be made into lasers too. These are indium gallium arsenide photonic crystal wires, and they basically laze. And you can actually get CW, room temperature lasing out of these structures. This is what the mode really looks like, and you see that the thresholds are pretty nice. They're on the order of 1 or 2 microwatts. So very low power you have to put in, and you get the material in you get a signature of the material around it. Now I can make a laser differently. I can say I have a fluidic system, and I might as well put some laser dye into it. I simply pump dye rather than water or saline solution or blood serum through it. And I simply take the laser dye, put it in here, and I have a replication molded channel. Well, that replication molded channel doesn't have to be a straight channel. It can have little bumps in it. If I put these bumps at the right correct spacing, I have a grating that forces the light to interact with it. And basically, I can make a distributed feedback laser out of the structure. This is now a replication molded channel with a DFB grating in it. And it has a certain wavelength at which the dye will start lasing. And that wavelength, in this case, is about 570 nanometers. It's because we use whatever the dye is. Okay? Rhodamine, I think. Rhodamine 6G gives us 570 nanometers. Rhodamine 101 gives different laser dye, gives us the red 610 nanometers, and so on. But this thing is also made out of rubber. It's elastomer. So I can simply stretch it. And if I stretch it, my laser frequency changes. I change the distance between these little bumps that I've put in. I can make a laser that is tunable. It's just a dye laser that has, is tunable mechanically, like a rubber band laser. Or I can lithographically change the difference between bumps, and I can make structures like this, where I have specific distances between lasing frequencies. I can take this and stretch it, and then tune each one, too. So, sort of interesting, because you can make structures that typically cost tens of thousands of dollars on a laser table, and you can make them for $1 or less, right? So that's what we think of at least what I think of when I think about optofluidics. I think about optics and fluidics connected together in an intimate way, all right? I'd like to talk about medical applications next. I want to think about how do we actually solve some of the problems that occur in the clinical world. Well, some of the problems look like this. This is a big instrument that's a blood test system. It basically does all sorts of things. It's like a little robot that moves around sample, prepares them. And we want to build systems that look more like this, a handheld unit that allows us to do the test. So we can build fluidic systems that allow us to do this. So one of the systems is shown here. This system has about 600 or 700 valves on it. It's about 1.5 inches in dimension. And we basically can do tests based on moving around solutions. So we move solutions around pneumatically in this case. The pink lines here are the flow channels, and the blue lines are the control pneumatics that allows us to move solutions around. We load this up with some antibody, different antibodies in different channels, and then we put our sample in the perpendicular channel. So basically, by opening and closing these valves, we can either have channels that conduct through like this. That's how we load the antibodies. Then we put our sample in. It goes in the perpendicular direction. And we can pump the sample around. So I now know the volume of my sample. The volume is about 0.5 nanoliters. And I can move that 0.5 nanoliters over my surface functionalized surfaces many, many times and collect all of my proteins on them. And after that, I can finish the sandwich assay by putting a dye on top of it. And I can look at the fluorescence coming out of these intersections, and that tells me something about the concentration that I had. I can actually see a linear relationship between that fluorescence and the concentration, and I can measure things in a couple of tens of picomolar concentrations. Different proteins. VEGF is a cancer indicator. PSA is prostate cancer indicator. CRP is C-reactive protein. It's a cardiovascular thing. So you can do many different things now, and you can make them in a very compact measurement system. You can go further, and you can integrate magnetic devices. And I've talked about that in the past, but I'm not going to dwell on it. But basically, these kinds of systems can be integrated in many different ways to read out protein levels. Now I'd like to talk a little bit about a project that is very dear to me, which we're just starting together with pathologists at Dartmouth. It's a project where we amplify DNA. So the team members are myself, Professor Baltimore, who's been working on RNA and DNA for a long time, and Professor Tsongalis, who's also at Dartmouth Hitchcock Medical Center. We've started a project that's supported by the Bill and Melinda Gates Foundation to try to do very cheap and inexpensive molecular diagnostics in a third world environment. We use PCR. PCR is a mechanism with which we can amplify DNA. It requires us to heat and cool the DNA to split the DNA into single-stranded molecules and then replicate it many times. So typically a heating and cooling system looks like this. You heat up, cool down, anneal, heat up, cool down, anneal. Do that many times. Each time you do that, you double the number of molecules that you have. So you would have 2 to the power of however many cycles you have here of amplification. Well, one of my students wanted to buy a Pcr reactor about 5 or so years ago. And I realized that they cost $50,000. So I said no. So he built it. Turns out that it looked like this. It's not very pretty, but it works. He built it for about $800 in parts. Of course, his labor, we don't figure that. But in any case, what's interesting is he built something very unique. This had liquid metal cooling, so that was extremely fast. And it allowed us to do amplification not in 45 minutes, which is typically what you get in a real machine, but within 65 seconds. Okay? Now the problem is, at this rate, the enzyme doing the amplification couldn't keep up with our system. So we had to actually slow it down. We actually had to go down to about 100 seconds before we saw real amplification. All right? But it's neat, because now we can tell the biologists to speed up their enzymes. Okay? And then the student went to Bangkok to try it out. So we found a virologist with all sorts of nasty things in his refrigerator. And he was willing to compare what he used, which was a Roche light cycler, with what we built. And we basically took these tests, basically indicating that our system did the same kind of clinical quality work that he had, except that our system was about 10 times faster. Okay. So that sort of grabbed my attention. I said, well, we should continue this. We should figure out how do we make this now even less expensive and even better. And we started thinking about different technologies, and this is one of them. This is using an electromechanical relay to heat and cool things. This is the machine that we're building at the moment. And it costs about $200 in parts. It basically has a disposable, and it allows us to do 100 reactions with a AA battery. The heating and cooling is really fast because it has a metal plate underneath it. The metal transmits the heat. And it's lithographically built, so I like it. It basically allows us to control the cooling rates and heating rates. And we can get now, within 6 minutes, we can do the PCR reaction. We can go one step further. We can build these kinds of systems. Instead of assembling them together and building them in little modules, we can start building PC boards and putting them together. So let me talk a little bit about PC boards. One of the biggest problems in molecular diagnostics used to be the access to this expensive equipment. But it turns out now we've removed that problem. We can make this instrument for $200. We've created another problem. The bottleneck now is, how do you make the sample for this instrument? How do you actually prepare the sample? And how do you automate that? Because you need a very skilled person to prepare the sample. Every sample is different. Blood sample versus urine sample versus other samples. They're all different. Well, that got us into thinking that we should build these systems, again, by printing. But in this case, by printing with larger dimensions than on a chip with PC boards. PC boards are readily available, and you can integrate them with other technologies to build structures. Well, in order to prepare a sample, if you had, say, tuberculosis, you want to first lyse the system. You want to take the bacterium and you want to open it up so that you can figure out how to get at the DNA. And a typical lysis system looks like this. This is called a French press. It's not a very subtle instrument. You heat it, you heat up and you press together the bacteria to a pressure of about 40,000 psi. This is very difficult to miniaturize by printing, all right? But what we can do is we can do something else. We can print channels where our local pressure is about the same as the French press does in a global way. So we can have a bacterium come in here, or a cell come in here, and smash it in a flow channel, okay? In fact, we can go one step further. We can put a little knife edge right in the middle of the channel. We can smash it and cut it apart at the same time. And so we build these systems by printing. We basically can build systems that allow us to smash even yeast cells, which are sort of the same as tuberculosis cells in terms of their cell wall and durability. If we can do that, we actually don't need much pressure. We can just have a syringe, put in the sample, and it destroys it and lyses it. This allows us to really build systems much more compact. Okay? So we have a system now that where you filter out the cells, you can lyse the cells, you can do collection of DNA, and then you can do PCR. While PCR can also be done on the PC board. You can actually have a little chamber, and you have some heater underneath it, and you can build systems like this. We basically have a PC board that can give you a Pcr reactor, and that can cost maybe $2. Okay? So basically, the idea is that we're going to a world where we will have these handheld battery-operated systems, and we're going to make them by printing. Okay? And we'll hopefully be able to solve some of the problems in second world countries and third world countries and figure out how to provide medicine to people that need it. Well, we'll talk a little bit about where we're going to go from here. We've built very complicated fluidics. This is maybe our most complicated chip. It's a single cell analysis chip. It allows us to take 30 cells, individually put them into little chambers, and then compare the RNA libraries and gene expressions from each one of these cells by doing a PCR reaction on the other end, extracting the RNA, doing a cDNA conversion, and doing all of the rest of the process over here. Okay, this is what this chip looks like. These are the cells somewhere inside of these chambers. There are 32 of them. And then there's the valve. You open up the valve, and then you pump around the lysis buffer around. So this is what the system looks like. Turns out that the student had coffee, so it's a little jerky. Basically, you can see now you start pumping it, and the buffer goes around, and then you extract the RNA over here on the next step, which is essentially a bead column. This is the bead column. Anyway, I won't bore you with this. But basically, the system looks like this when it's all done. It has all of these tubes coming out of it because they're pneumatic lines that control all of the 2,000 valves that are on here. This is a chip, a microfluidic chip, with a huge relay rack next to it in the laboratory. Okay, we want to go one step further and say we want to build a laboratory without the relay rack on the chip. So we want to remove all of this hair of Medusa type of connectors. Okay, we want to build this in our way. And we're actually doing that by adding new valve technology and so on. We think we can do this all and build handheld systems, but where do we go from there? Well, we think that we can go one step further. We can actually take little chips that are small enough and have a single function or maybe multiple functions that are small enough to be implanted into systems. Now, this could be into the fuselage of an airplane, if it's a graphite reinforced epoxy, or they could be in a person that suffers from diabetes and you want to figure out the glucose level. In both cases, you don't want to have a big thing. You want to have a little thing. So we can build things now that have this function at the scale of 100 microns or so. Okay? The way that we do this is by powering right on the chip and by sensing something from the chip, having some amplification circuit, and then transmitting the data off the chip back into a reader. The idea is to make it completely wireless. Well, wireless in that it's not connected to the rest of the world. That means that you don't have to worry about infection or anything like that. And I should mention this is work that we're doing together with another group at Dartmouth, Sol Diamond and Eric Kobilars. So the idea is you make this little chip. It has a little transponder in it. It's got a power supply. Power supply is a photovoltaic. You have a little circuit, amplification circuit. You have a laser that transmits the light back out, and a bit stream of light will tell you what's happening on the detector. This is what it looks in schematic. Maybe, as Sol wants to do, you want to hook this up to a neuron. You basically have a system that's attached to a neuron, has an amplifier here, and then you get the photovoltaic to power that circuit and to power then the transmission of data from that amplifier to the outside world. Well, you need to think about a couple of things. First of all, you want to operate at a wavelength for the photovoltaic as well as for the light emitter where you don't absorb the light. And for the body, the optimum wavelength is somewhere in the near infrared, because that's a window at which you minimize the absorption losses. And I'm not worried about absorption losses because I don't have enough power in my laser. I'm worried about absorption losses because I'm going to heat up the tissue, and I don't want to heat up the tissue, right? I don't want to boil the cells while I do the experiment. So the power supply could be just a little pin diode, or it could be something like this, a laser or a silicon photovoltaic array. In this case, these structures are very small, so you can make them down to 50 microns or so in size. You can get reasonable power out of them. And then you can basically take this, this signal and transmit it out by using another laser. In this case, we use a vertical cavity laser. It's a conventional device that's been optimized over the years and basically has a pretty low threshold current, so we can work at low powers. You can make the device so that each device has a specific wavelength laser, so you can transmit data with very large bandwidth, measure many devices at the same time. Okay. The kind of things you could measure are diabetes, maybe these ions for dialysis patients, maybe some cancer markers, maybe you want to measure neurons for neurologic disorders, Parkinson's disease, or maybe epilepsy patients, and so on. The kind of system the way that it works is you take whatever you measure from your circuit, you convert it into a bit stream. That bit stream corresponds, in this case, the density or the frequency of the bits corresponds to the voltage that you measure. And you can then transmit that out and read it on the other end. You can have all of these different detectors. So now I'd like to sort of come to the last part of my talk. The last part of my talk, I'm going to show you sort of the extreme smallest structures that we can make. It's particularly interesting for me because I like very small structures. And it turns out that we can think about taking the size and the geometry of a structure and determining the function. All right? In the past, if we want to have a different band gap semiconductor, we just look it up and get the chemistry right, and we grow it. Right? I'm going to suggest that there's another way, that we can take the band gap that we want, and we decide what geometry we need to generate that band gap, and we lithographically define it. So let me show you how that works. So we can make small structures. In fact, we're not the only ones that can make small structures. Intel makes wonderful small structures. If you buy your chips nowadays, you can get 32 nanometer gate lengths, so pretty small. So these are 40 nanometers, so not as small as Intel's 32 nanometer structures. We can make them, in this case, by electron beam lithography. This is not a manufacturable process, but we don't need to manufacture it. We'll give it to Intel when it works, and they'll make it very cheaply. In fact, Intel has a roadmap. This is their roadmap. We're now at P1270. Okay, it's 2011, so we must be on this part of the roadmap where we're going towards 22 nanometer features on the chip, on a 300 millimeter wafer. Okay, all over it, perfectly aligned. That's what Intel does. The transistors, this is the gate length. The transistors don't look like this anymore. They look like that. They're becoming more and more three-dimensional because of the small size. You have to do this. So where do we go in the year 2020 off this roadmap? Well, Intel says we're at 10 nanometers in 2015. Well, it turns out we can make structures down to 2 or 3 nanometers in my lab. So we can make structures to see what happens when we get to 2020. This is the kind of structure that we can make. Instead of a transistor that's in plane, this, by the way, is a structure that has enabled all of the miniaturization of circuits, really. It's a DRAM structure. It's actually a MOSFET. And it hasn't changed much over the last, say, 40 years. It's remarkable. A single device hasn't changed in geometry, just shrunk in size. And it's revolutionized how we think, how we do things. So what's neat now is that for you guys, the new device engineers, you don't have to worry about this structure anymore. It's going to be replaced at some point with something else, something new. And maybe the something new will look more like this. It's now a channel that's in the vertical direction, and it has a gate around it. Okay? It's the same device still, really, functioning roughly the same way, with some differences. So we can build little pillars like this that are maybe 10 nanometers in dimension. Right? Intel will be able to build these in 2015 in a 300 millimeter wafer. We can build them now in small quantities. But let's say you want to measure it. So you have one of these structures and you want to measure it. Well, this is what it looks like. Here's our probe tip. I would call it Godzilla, or Godzilla's foot maybe. And you see the structure. This is the silicon. You start hitting it. Of course, this is not that accurate, right? So you push it down a little bit. The silicon bends. Normally, silicon is this really brittle material. But at this scale, you start seeing different mechanical properties. It's bending. And then when you take the probe tip off, it snaps back. In fact, if you go to the extreme case, in this kind of bending experiment, you see this little silicon whisker is almost bent perpendicular to its original orientation. And then in the next frame, it snaps back perfectly. It's 25% elastic deformation. Okay, so mechanically it's different already. Now we can think about what do we do if we want to make electrical measurements? Well, we can oxidize it. So we start with something that's maybe 10 nanometers or so in size, and we oxidize it. We make it smaller, shrink it down. So the silicon now is in encapsulated by an oxide layer. And what's interesting is this oxide is denser and thicker and bigger than the silicon. So the silicon starts and it forms the oxide. The oxide is bigger, so it'll put the oxide under compressive stress and the silicon under tensile stress. And at some point, this oxidation process actually stops because there's no oxygen diffusion through this compressive silicon dioxide. And you can make structures that are very stable, that have silicon pillars within oxide encapsulants. Well, you can make them as small as you want to, really. Well, maybe you can't go much below 2 nanometers. Okay, in this case, this shows you the diameter of whatever pillar you get as a function of the temperature that you've oxidized at. You can leave this in the furnace for 5 hours or 10 hours. It's okay, because the diffusion stops. Okay? And you can make whatever size structure you want by dialing in the temperature. So at 3 nanometers, you would dial in, I don't know, some temperature over here. You basically end up having this capability of making smaller structures than you lithographically started with. And these smaller structures behave differently. So if you look at them by doing photoluminescence, you actually can see that the energy of the luminescence is going up as you shrink the device. So if you go from 8 nanometers to 2.5 nanometers, you see this shift of the band of the luminescence. But wait, silicon shouldn't be luminescing because it's an indirect bandgap semiconductor. It turns out that you've changed the band structure. You've actually changed the band structure so it becomes direct because you've made a very small whisker and you've put it under tremendous tensile strain. Okay? You can calculate the effects by using tight binding models and so on. So this is very interesting because now you've made a material that's chemically silicon, but geometrically you've changed things. You've introduced basically a change. And you can measure this. All right. So what's the opportunity? First of all, if you have a high band gap, it means your device will work at higher temperatures. So that's probably a good thing. If I want to have my laptop work at 200 degrees centigrade, maybe that's a good thing. Okay? Don't have to cool it as much, maybe. I don't know. But the most important thing is you can design the band gap, and you can do the band gap engineering. So you can make a structure like this, where you change the band gap here, and here, and here, depending on the geometry. Okay? So how do you do that? Well, you can etch the silicon, and you can undercut it. But if you correct that, you can go and put a dimple in it. Okay? This is now 3-dimensional. You've made a 2-dimensional object that with a very small mask, but you've now made it 3-dimensional by changing the etching conditions while you're transferring the pattern down into the substrate. You can make a corrugation. It goes in and out. Okay? You can make this corrugation as wide as you want. And then if you oxidize that, you end up having little dots, little quantum dots. And these quantum dots, again, show you the blue shift. In this case, you can see what I like about this image, this figure is, you see the difference between a 2 nanometer and a 2.4 nanometer silicon quantum dot that you've made. You've added 1 atom, okay? And you can see it. It's really cool. And so basically, the whole idea is you can now make these kinds of nanostructures, essentially 3-dimensional nanostructures, by doing a combination of lithography and careful etching. Okay? This is a really neat direction, I think. One of the problems of this is if you make these structures so small, you can't dope them anymore. Because if you look at it, statistically, the dopant concentration, if you dope with 10 to the 19 dopants per cubic centimeter, and you calculate how many you should have in a 2 nanometer structure, you'll end up with a couple. Okay? It doesn't give you enough statistics to have a Fermi level and do the p-n junction thing that we're so used to. Okay? So we need to introduce carriers by gates. We have to build structures like this that have 2 or 3 gates on them. Okay, and when we build those structures, it's tough to build them, but when they work, this is how they would look like. This is a TEM cross-section of one of the MOSFETs, and this is how they work. So we can make actually reasonably good transistors by making these 2 and 3 gated structures and building them. We can also make sensors. We can make ion sensors, where this kind of gate can then absorb whatever ions are surrounding it. And you can measure the concentration by looking at the voltage at which the channel turns on. So it's a nice little ion sensor. The dimensions are 15 nanometers for these. You can package them. This is what they look like when they're packaged. So there's a great opportunity, I think, for miniaturizing through lithography. I think geometry, controlling the geometry, is going to become increasingly important as we go down this road of miniaturizing. We can think about miniaturizing electronics. That's what everybody does and has been doing. We think about miniaturizing optics. We can think of miniaturizing fluidics. All these systems can now be put together. We can make them with the same alignment marks, and then we can align them on top of one another and build multifunctional systems. I'd like to sort of thank you, but I'm going to stop after, well, I don't know, do I have 1 minute?
Sure.
I'll show you a lithography experiment. This is me. This is the microscope. I love microscopes because I'm fascinated by small things. This is what makes me really happy. Instead of shuffling paper, I sometimes have the freedom to go into the microscope lab. You have one of these. It's a TF20. It's beautiful. Anyway, this is what you look through when you're looking through a microscope. You have an electron microscope, so this is an electron image. You focus it down to a small spot, and then you look at it and defocus it, and you see you've actually made a small hole. Okay, and it becomes apparent if you measure this. It's actually 2 nanometers in size, and it's about 100 nanometers deep. Just done that. So I'd like to thank you for your attention.
Any questions from the audience?
How reusable are these fluidic labs on the chip? Are they intended to be disposable, or I guess they're cheap?
Yeah, the question is how reusable are these little labs or microfluidic systems? And I would argue they're probably not. Especially the elastomeric ones are very difficult to clean without destroying them. But since they're so inexpensive, you can throw them away. That's the concept, at least. Having said that, the key thing is naturally, if you're looking for pathogens, you don't want to have any chance of having false positives. And so therefore, I think it's prudent anyway to build systems where you minimize the risk of contaminating samples. So I would say for molecular diagnostics, and especially when you're amplifying things, you would want to dispose just even if it was possible to clean them. Okay.
How actually do you manage to modulate the etching as you go on the filler?
Yeah, it's something we've been trying to do for quite a while. We used to make vertical cavity lasers in the 80s when that was a cool thing to do. And basically, we would change the etching conditions to make sort of a little hourglass structure. And so you do that by changing the chemistry during the etching reaction. You increase the gas pressure a little bit, and you increase the power a little bit, and then you decrease the gas pressure, and you come back out. So you can do that, and we've been doing that with a resolution of, say, hundreds of nanometers in the past. But my student figured out how to really control the modern reactors, which are automated. You can actually program in what they do. You don't have to sit there with a knob anymore. And so that's enabled him to get this very precise control on the order of 10 nanometers. And so it's all about the gas chemistry and the power levels in the reactor. And the precision with which you can do this is nowadays adequate to do these kinds of structures. This is, I think it's a really neat way of making a heterostructure out of a single material. Yeah.
When you're making these very small structures, what does it take to build the mask that you use to make them?
Ah, yes. When we go to the smallest structures, what do we use to make to make the mask? Well, we use electron beam lithography in my lab because that's convenient for us. We don't have a projection mask aligner with a 22 nanometer resolution. However, in commercial approaches, and the 32 nanometer node, and now more recently the 22 nanometer node, it's all done optically by using what's called phase shift masks. Now, the masks are pretty expensive. They cost maybe $50,000 or so, or upwards from that, because they have dielectrics to make sure that your intensity on your sample is optimized. You also, I mean, this whole area of it's remarkable that you can make structures that are so much smaller than the wavelength that you're making them with. But it's possible, so the technology is there. And what we do is simply, we refer to the fact that people are doing this, and that makes it manufacturable, especially in silicon. So if you make something in silicon that works, at any of the structures that I've shown you, we start with relatively large features, huge features, 30 nanometer features, right? We start with those, and we make them into 3 nanometer features, right? The neat thing is, Intel can do the very same thing today if they wanted to, right? Because they have the technology to do this. And so if we get something to work properly, then it can be directly manufactured.
I invite you to speak with Axel if you have more questions. Thank you very much.